Hello my dear students, welcome back to my channel, and today we are going to discuss about the topic DNA packaging. So let's get started. What is DNA? DNA or deoxyribonucleic acid contains all the information that are necessary to build an organism and to maintain life. DNA carries the genetic code and the DNA code contains instructions that are needed to make proteins that are necessary for our growth, development, reproduction, etc. Genetic code is a set of instructions that directs the translation process. So, DNA first replicates to make copy of itself. By using a strand of DNA as a template, pre-mRNA is formed and it takes in the information from DNA. This process is called transcription. Then the pre-mRNA is processed to produce mature mRNA, and the instructions that mRNA carries are used to assemble amino acids in the correct order to make specific proteins. And this process is called translation. We all know that the amino acids are the basic structural and functional unit of protein. DNA is composed of two polynucleotide chains that coil around each other, and it looks like a double helix. DNA is made up of nucleotides. Nucleotides are the building blocks of DNA. The nucleotide consists of a sugar molecule, a phosphate group, and a nitrogen containing base. The sugar molecule is attached to a phosphate group and a nitrogen containing base. The bases are of two types, purines and pyrimidines. There are two types of purines, adenine and guanine, and two types of pyrimidines, cytosine and thymine, are present in the DNA. The two strands of DNA are held together by hydrogen bonds between the bases. Adenine binds to thymine and forms base pair, and guanine binds to cytosine and forms base pair. What is the meaning of DNA packaging? DNA packaging is the folding of DNA into a compact structure. It is the process by which DNA is tightly packed and fit properly within the cell nucleus. Why is DNA packaging important? The DNA is actually very long, and a large number of DNA is stored inside the cell. Let's take an example. In case of human body, each diploid cell contains 23 pairs of chromosomes. That means total 46 number of chromosomes are present inside one or a single diploid cell of human body. The chromosomes are made up of two strands of DNA. So, the number of DNA in each diploid cell is 46 multiplied by 2 is equal to 92. And each haploid cell contains half number of chromosomes, that is 23 chromosomes, and the number of DNA strand in each haploid cell is 23 multiplied by 2 is equal to 46. If we stretch out a DNA, it would be very long. It would be more than 6 feet or more than 2 meters. But the diameter of the human cell nucleus is approximately 10 micrometer. So, each DNA has to be packed and wrapped around histone proteins to fit nicely inside the microscopic nucleus. Histone proteins play a very important role in the process of DNA packaging in eukaryotic cells. Most prokaryotes do not have histone proteins at all. Prokaryotes compress their DNA into smaller spaces through supercoiling to fit inside the cell. Researchers found that multiple proteins act together to condense prokaryotic DNA. and these proteins are the non-histone proteins. One of the major proteins is HU protein that is present inside the nucleoid of bacterial cell, and it works with the enzyme topoisomerase or gyrase and helps the DNA to fold or to condense. The gyrase protein has an important role in DNA replication. We will talk about this later. That's all for today, students. We will continue this in my next video. Stay connected with me and thank you so much for watching my video.